Welcome to Freeslate's webinar on automated analysis of protein formulations. This is the 8th webinar in our ongoing series. My name is Justin Fisher, and I'm the Hardware and Software Product Manager at Freeslate, and I'll be your host for today. Today you're going to hear a presentation about our family of CM Protégé systems for bioformulation analysis. We're pleased to have Dave Yamani from Freeslate joining us. Time permitting, we'll conclude this webinar with a brief question and answer session at the end. Before we begin, let me just invite you to submit questions using the Q&A menu on the right-hand side of your audience console. In the Q&A window, you can type in your questions and press the Submit button. Any questions that we do not get to will be answered privately in a follow-up email message. Our speaker today is Dave Yamani. Dave is currently the Product Marketing Director, Life Sciences at Freeslate, where he manages the Life Science business segment. Prior to Freeslate, he was Senior Director, Drug Discovery Marketing at Molecular Devices, where he managed a drug discovery line of business consisting of 6 products. From 2005 to 2008, David was Director of Electrophysiology Marketing for Molecular Devices. Prior to that, he's held other marketing, sales, and support roles, where he's been responsible for bringing a variety of life science products to market. The title of Dave's presentation today is Automated Analysis of Protein Formulations for Visible Particle, Turbidity, Color, Viscosity, and pH.
Good morning, everyone, and thank you for attending today's webinar. I'd like to welcome you to today's presentation, Automated Analysis of Protein Formulations. And for today's presentation, I'll start out with an overview of Freeslate. Then I'll go on to tell you about the CM Protégé family of bioformulation assessment systems. And then I'll end today's presentation with a couple applications, in particular, high-throughput viscosity measurement of protein formulations, and the analysis of formulation samples for an accelerated stress study. So just to give you some background about Freeslate. So originally, Freeslate is a spin-out from Symyx, and originally Symyx had 3 businesses. They had a software business, an R&D business, and a custom automated tools business. And a few years ago, Symyx decided to focus on their software business, and as a result, they spun out the tools business, and that spun out as Freeslate. When Freeslate was spun out, it retained the Symyx's IP on high throughput and microscale technology, and so we have over 16 years of IP in this area. We have a broad global customer base with sales in the U.S., Europe, and Asia. We have approximately 100 employees, and about 60 of those are scientists, engineers, and developers. Our headquarters are in Sunnyvale, California, and we have an office in Geneva, Switzerland. We have over 100 customers in the U.S., Europe, and Asia, and we have over 15-year track record of delivering automated solutions to the customers like you see in the right-hand side, such as Johnson & Johnson, Amgen, Pfizer, AstraZeneca, and Bristol-Myers Squibb. For our business, we actually concentrate on 2 verticals. One vertical is chemical and energy, which you'll see on the left-hand side. And in that vertical, we focus on delivering automated solutions for catalytic transformations of biomass, organic molecule, catalysis synthesis, and automated solutions for polyolefin catalysis and process development. The other vertical that we focus in is drug development. And in that vertical, we're working on solutions for doing protein therapeutics, which we'll be talking about today, for protein formulation, protein purification, and also for small molecule therapeutics, such as automated solid form screening systems, polymorphin salt screening systems, force degradation, process optimization, and automated systems for doing organic synthesis. So we have a full range of solutions. You can see the range of solutions in the slide above. So for customers looking for complete automation of complex workflows, we sell highly specialized customized systems like the one on the left-hand side. And the system on the left-hand side is a CM3-based system, which is our bioformulation workflow. And for those types of systems, it really is full automation to support customers that have capacity constraints. And typically those installations have physical integration of the analytics on deck. And for example, on the bioformulation workflow, it has an integrated dynamic light scattering instrument and UV spectrophotometer. The complexity of those workflows are typically high, and the profile of the user is usually an automation expert. The next type of workflow that we sell is configurable workflows. And those configurable workflows are based on established workflows that we've developed already. And so typically the configured system is just a slight modification of that standard system that we have. And so those really are developed to support automated workflows where there's higher throughput is required. Typically, instead of having integrated analytics, they'll have virtual integration analytics. The complexity of the workflow is medium. And typically those systems are operated by trained scientists and technicians. The third type of workflow we have, which are the ones that we'll be talking about today, are workstation-based workflows. And these workflows are easy to use and really are designed to address specific assays used in many labs on a day-to-day basis. For those, you can purchase virtual integration of the analytics, which is optional. Usually the complexity of the workflow is low, and those workflows are walk-up type instrumentation. So the solutions that we deliver are really based around our proprietary automation platforms, our laboratory execution analysis software, which we call LEA. And LEA wraps around our products and really creates a seamless integration from the design of the experiments all the way through to the data analysis and data management of the results. And then to ensure that our customers can extract the most benefit out of our high throughput solutions, we have our scientific expertise and collaboration with our customers to make sure that they're successful. Well, biologic formulation development is really the most critical and challenging aspects of biologics development. So the ideal formulations will maintain the long-term stability of the protein. They'll be compatible with the chosen mode of delivery. They'll meet the regulatory requirements. They'll enhance the drug manufacturability, and they'll ensure clinical performance. But there's many factors that can affect the integrity of the protein in the biologic formulation. And some of those factors are pH and buffer conditions. There's temperature, oxidation, agitation, and exposure to surfaces. So proteins are really very challenging molecules because they can degrade in many ways, and they're very, very complex. And so some of the ways that proteins can degrade is chemically. And so proteins can undergo deamidation, which affects the asparagine and glutamine amino acids. And the rate of deamidation is affected by the protein sequence, the pH, temperature, and ionic strength. There's also proteins can get oxidized, and that typically is of the histine amino acids, methionine, cysteine, tryptophan, tyrosine, and potential oxidants are hydrogen peroxide, UV light, metal ions, DMSO. And proteins can also degrade physically, so they can go through subunit dissociation, denaturization or unfolding, particulate formation, or aggregation. And aggregation turns out to be one of the primary pathways to degradation. Aggregation can occur under both physiological and non-denaturing conditions, and typically if your protein aggregates, it's irreversible. You can't reverse that. And aggregation is affected by pH, the ligands and the co-solutes, salt type and concentration, the presence of surfactants and preservatives, and agitation. And all those modes of degradation will lead to potentially an unstable product and a short product life. So because there's so many different ways that proteins can degrade, and proteins are so complex, there's many different assays that people use to characterize their proteins. And so I just simply compiled this list of the different techniques that people use and the endpoints that people are looking for. And so this is just a representative example. And the Freeslate decided to focus on pH, viscosity, color, particle counting, and turbidity because those assays are predominantly all manual, and they're typically performed in a serial fashion on individual instruments. And so what we did is develop a platform that would take these different assays and automate them. So the goal was to come up with a solution that really frees up scientists from these assays so they can actually do more productive things. And that solution that we came up with was the Protégé Bioformulation Assessment System. Well, the Protégé Bioformulation System automates visible particle, turbidity, color, viscosity, and pH measurements of protein and vaccine formulations. It enables scientists to screen samples quickly to support formulation screening campaigns, allows them to rapidly analyze formulations from accelerated stability studies. And since the system can operate unattended, It means that scientists can analyze hundreds of samples per day to complete projects faster and explore a broader formulation space. So the Protégé bioformulation assessment system is really a family of systems. And so there's actually 3 different configurations. There's the 200 system, which consists of visible particle counting, turbidity, color, and pH. There's a 300 system, which has the capability to measure viscosity and pH. And then the 400 system, which has the capability to measure visible particle, turbidity, color, and viscosity. Just as an example of the deck layout for a Protégé system, we have a picture here of the Protégé Bioformulation Assessment 400 system. And so what you can see in the upper left-hand corner is the vial plate gripper and flexible tool changer, which we use to move plates around the deck. You can see on the left-hand side a waste bin where the pipette tips get disposed of after they're used. You then see the on-deck viscometer that we use for viscosity measurement, a tool and plate rack, a 3-position plate rack, and then the visual inspection station where we do particle counting, color, and turbidity measurements. And so since the system is designed to operate unattended, we wanted to add the capacity so people could load up the system with many plates. And so then on the far right-hand side, you see a vial plate hotel. For people that are interested in having vortexing on the system, we have an optional on-deck vortexer that can be added. And so the on-deck vortexer is pictured here, and the vortexer holds 3 different plates. You can see the 3 different plate positions going from front to back. And so the plates that the system holds are standard microplate format. And so what you would do is get a microplate, get a plate holder with the size holes that you need for your different vials. And so the vortexer can accommodate many different sizes of vials and plates. For systems that measure pH, it comes with a 4-channel pH probe arm element. And so for that pH probe, we have 4 solid-state-based pH probes. And the advantage of using a solid-state pH probe is that they're very, very robust. And it's a solid-state sensor, so there's no glass electrodes. The throughput of the pH arm element, it can read an entire 96-well plate in 48 minutes. And the resolution is about 0.1 pH units. It has an integrated temperature sensor. And really, the solid-state pH probe overcomes issues of traditional glass electrodes. And I remember when I was in the laboratory that some of the issues I ran into was that the pH probe was drying out, they're unstable, and they're prone to be getting fouled. So we did a comparison of the solid-state pH probe against the glass pH electrode, and we did that comparison using pH 4 buffer, calibration buffer, and pH 10 calibration buffer. And so here you can see the results of that comparison. You can see that the average pH measured with both solid-state and glass microelectrode are very similar, but you can see that the reproducibility of the pH 4 buffer is much better with the solid-state electrode versus the microelectrode. And also you can see that the number of readings that were outside the acceptable range is much lower with the solid-state electrode than with the glass electrode. If you look at the pH 10 measurement, again, you can see the average pH values are very comparable between the solid-state and microelectrode. And again, the reproducibility of the solid-state electrode is much better than the microelectrode. And also the number of out-of-spec or outside of the acceptable range data point is much lower with the solid-state electrode. But the biggest difference between the solid-state electrode and the microelectrode is that with the solid-state electrode, you actually have a 6x reduction in the measurement time of measuring the pH. And so you can get much higher throughput using a solid-state pH electrode than you can with a glass electrode. For systems that measure viscosity, the Protégé Bioformulation Assessment System has an on-deck viscometer. And the viscometer that we develop is a high-throughput capillary viscometer that uses positive displacement pipette to inject the sample. The viscometer can measure viscosities accurately up to 100 centipoise. It has temperature control between 4 degrees C and 40 degrees C. It has an integrated automated wash system to ensure that there's no carryover between samples. And the viscosity measurement is very, very quick. It's less than 3 minutes per sample, and it only requires about 100 microliters to measure your viscosity. So to benchmark the performance of the high throughput viscometer, we measured 4 viscosity standards. We measured at different temperatures. We measured a 1 centipoise and a 99 centipoise viscosity standard at 4 degrees C, and a 36 centipoise and a 0.7 centipoise viscosity standard at 4 degrees C. And you can see below the results of that comparison. And you can see, if you look at the data, that the reproducibility and accuracy of the high throughput viscometer is very, very good. For visible particle, color, and turbidity measurement, we developed an element called the Visual Inspection Station. And typically, color and particle detection are all done manually, and they're done by an actual human looking at the individual vials and trying to ascertain whether there's particles in the solution or the color of the solution. And so it's a very subjective type measurement. And so by automating that process with the Visual Inspection Station, we actually take that subjectivity out and make it a very objective measurement. And so the way that it works is the gripper arm lowers the vial into the Visual Inspection Station. You can see that there's a camera behind the vial, and there's an illumination source in the front of the vial. And so what we do is we use imaging to do the particle and color and turbidity analysis. So we use image analysis software, so it's a very objective and consistent measurement that we make. No specialized training is required in order to do the analysis. Because it's automated, you can have unattended operation. All the images are stored for reanalysis and review, so if you have to go back and relook at an image or reanalyze data for any reason, that isn't an issue. It's higher throughput than manual methods, and it eliminates human interpretation error because all the data is coming from the image and the software analysis. And the analysis is non-destructive, so we analyze the samples directly in the vials. So the way that we do the color analysis is really a color matching process. And so what we would do first is we would measure the calibration or color reference solutions, and in this case we use the European Pharmacopoeia BY standards. And once we measure those, we then measure the sample color in the samples, and we then compare the color in the samples against the calibration standards to ascertain the color standard that it best matches with. And so an example of doing that analysis is this analysis of 5 monoclonal antibody solutions. And across the top here, you can see the calibration standards from BY1 through BY7. And you can see that we were analyzing 2 different monoclonal formulation concentrations, a 210 milligram formulation and a 50 milligram formulation. Actually, it's 3, and a 10 milligram formulation. You can see for Vial 5, the color matches very well with BY2. It had a match score of 84.8. For Vial 4, you can see that it matched very well with the color standard BY3. It had a color match score of 98.4. For Vial 8, it had a very good match score with BY4 of 91.4. And those are all the 210 milligram per ml standards. If you look at Vial 6 and 7, which are the lower concentration protein formulations, you can see that Vial 6 matched with BY4 and BY5, so the color is somewhere between those 2 standards. And you can see that Vial 7, which was a very, very faint color, matched somewhere in the range of BY5 to BY7. For turbidity, the process we use is very similar to color. So the first thing that we do is we acquire images of the calibration standards, and the range of the Freeslate turbidity system is from 0 to 1,000 NTUs. We then analyze those images, determine the average luminescence values, and we create a calibration curve. We then analyze the samples, and what we do is we analyze the sample and then subtract out the luminescence from a blank vial so we can subtract out any effect of the vial. And then we calculate the turbidity of the sample against the calibration curve. So here you can see an example of 3 monoclonal antibody solutions, formulations that we measured the turbidity of. You can see that for the image in the upper left-hand corner that we measured turbidity of 36 NTUs. We can see the turbidity of the lower left-hand side of 98 NTUs. And we can see that the turbidity of the sample in the upper right-hand corner is 181 NTUs. For particle detection, the way that we perform that is the instrument will drop the vial into the visual inspection station, and then it drops it in the nest and clamps down on it, and then we spin the vial very, very rapidly. And when we spin the vial very, very rapidly, any settled particles that are on the bottom of the vial get spun up into solution, and then what we do is we stop the vial very rapidly. And when we stop the vial very rapidly, we start taking images. And since the solution inside the vial is still spinning, the particles are carried around in the solution. And so you can see in the lower left-hand corner image of 2 consecutive images that we took, and you can see that the particles are, you can see the particle movement between those 2 images. So we take several images, and then what we do is we subtract the consecutive image pairs from each other. And the reason why I subtract the consecutive image pairs is that we can eliminate any kind of defects in the glass in the image by taking the difference image. We then analyze those image pairs to identify the difference image that has the highest number of particles in it. And once we identify that image, we then do a blob analysis on it to actually detect to count the particles. And the particle count is actually 1/2 of the particles that are detected in the blob image. So you can see the effectiveness of using spinning to suspend the protein particles that are in the solution. You can see in this on this slide. On the left-hand side, you can see the vial before we spin it with the protein settled on the bottom, and then in the middle slide, middle image, you can see the protein particles floating around after spinning. And then on the far right-hand image, you can see the blob image where we counted that there were 13 particles that were present in that vial. So as an option, customers can get the system with a full implementation of the LEA software. And the advantage of this is that with a full implementation of LEA software, you can also integrate data from your UPLCs, your UV spectrophotometers, and other analytics. And as a result of that, all the data from those instrumentation are dumped into the LEA database, which means that when you go back to visualize and analyze your data, that you can see both the data you collected on the CM Protégé bioformulation system, and you can see the data that was collected on a UPLC or UV spectrophotometer or whatever other analytic you integrated into the system. And so because you can see all that data in one consolidated view, it allows you to very rapidly build knowledge, which will enable you to drive faster decision-making about your protein formulations. So the first example of using the CM Protégé bioformulation system is one where a customer came to us, and viscosity was a bottleneck in their process. And so they wanted to evaluate the Freeslate Viscometer for throughput and also data quality. So the way they decided to do that was they sent us a protein solution of 221 milligrams per mil and a diluent buffer. And from the protein stock solution and a diluent buffer, we made up 13 different concentrations of protein standards. You can see those in the table on the right-hand side. We then measured the viscosity of those 13 protein solutions in triplicate at 3 different temperatures, at 5 degrees C, 15 degrees C, and 25 degrees C. The workflow that we use, you can see in the orange little arrows on the bottom. So first, we prepared the 13 target concentration, and we measured the pH at 5 degrees C. We then aspirated 100 microliters of the sample and measured the viscosity at 5 degrees C. Then warmed the samples to 15 degrees C for 30 minutes, and then aspirated 100 microliters to measure the viscosity. Then warmed the samples to 25 degrees C for 30 minutes and aspirated and measured them for their viscosity. The results of the viscosity studies were this. So the graph, you can see that what we saw was that the changes in viscosity align with expectations, which is what we expected, right? The viscosity solution increased with increasing protein concentration, and the viscosity solution decreased as the temperature was increased. But what was more important to our customer was the throughput of the viscosity system. And so using the Freeslate high throughput viscometer, we were able to complete 117 viscosity measurements in less than 1 day. We saw excellent reproducibility with RSDs in the range of 0.1 to 4%, and we really consume very, very little sample. So the analysis only takes up about 100 microliters of sample for each analysis. The second case study that I'd like to go over is analyzing formulations from an accelerated stability study. And so in that study, that was a collaborative work that we did with a biotechnology company. And the objective was to perform an accelerated stability study at 2 different protein concentrations in a variety of formulations. The formulations vary widely in pH, buffer species, as well as excipient type and levels. And so from the biotechnology company, we received 6 sets of monoclonal antibody formulations. And each set consisted of 12 formulations, which are F0 to F12, of a 210 milligram formulation of monoclonal antibody. It had 11 formulations, F13 through F22 and F24, of 50 milligram per mil monoclonal antibody formulation. And then we had a single formulation that was at 10 milligrams per mil, and the solution for that was saline. And the assays were performed using the CM Protégé bioformulation assessment system. So you can see how we did the study in the next slide. And so here in the left-hand side, you can see the 4x6 vial rack array and how we laid out the samples in the vial rack. And so one of the 6 vial racks went immediately to analysis on the CM Protégé bioformulation assessment system. And that was the data we used for our T0 analysis. We took the other 5 racks and put them into an incubator and ovens and a refrigerator. And so then we incubated the samples at 4 degrees C, 25 degrees C, and 40 degrees C, and then took them out at 45 days and did an analysis, and then took another set of samples out at 90 days to do the analysis. And when we did the analysis on the system, we were able to get turbidity, viscosity, pH, particle count, and color matching for all 24 vials, for all the time points and temperature points that we measured. So here you see the results from the pH measurement of the unstressed and stressed formulations. And the black line represents the design pH, or the pH, the target pH that the biotechnology said the formulations would be formulated at. What we observed was that the measured formulation pHs were a little bit different than the design pH that was specified. What we also see is that over the course of the study, that the pH was stable, except for very minor increases of the formulations that were stressed at 40 degrees C. But what's of note is that each time we measured the pH of a vial rack or a pH of the sample formulations at the specific time point, it required 72 pH measurements. And we were able to complete those pH measurements in about 36 minutes. With regard to viscosity, again, we measured viscosity of the samples at the different time points. And note the viscosity scale of the graph on the left-hand side is much bigger than the viscosity scale of the graph on the right. And so you can see that the 210 milligram formulation, that the viscosity increased dramatically over the course of the study. And that viscosities at the latest time point, at 40 degrees, 90 degrees, 90 day, exceeded the capability of the viscometer, which is 100 CP. On the right-hand graph, you can see the viscosity that we measured for the 50 milligram formulations, and you can see that the viscosity of those formulations is significantly lower than the viscosity of the 210 milligram formulation. And in addition, you can see that over the course of the study, that the viscosity of the 50 milligram per mil formulation showed only minor changes from the original measurement at T0. And so the Viscosity Station was able to measure the viscosity difference through the formulation, composition, and monoclonal antibody concentration, and the absence or presence of excipients. So for particle analysis, we only measured the very last, highest temperature, very last time point, which is the 45 degree C, 90-day set of vials. And what we found when we took the vials out of the incubator was that the high concentration formulations, the 210 milligram formulations, had like a gel-like consistency. And so when we put those into the instrument to measure the particles, the solution was too viscous to actually spin up the particles. And so we didn't detect any particles in those vials because of the solution's gel-like consistency. But the 50 milligram per mil solutions were just fine. And so we were able to measure particles in those. And you can see that formulation F13, we measured 4 particles. In vial F17, we measured 18 particles. F22, we saw a single particle. And F23 and F24, we saw 6 and 20 particles respectively. And so what I did is on the right-hand side, so you can see that in the unstressed vials that there weren't any particles present. There's images of the T0 measurement for the formulations that we detected particles in. And then on the right-hand side are the blob images from the analysis of those vials, where you can see that we detected particles in each one of those vials. The last analysis that we did was color assessment, and what you can see in this slide is the standards that we ran, which is BY1 through BY7. And what you can see in the analysis is that the 210 milligram per mil formulations, which were from F1 to F12, that the color was predominantly in the BY3. And we had very good matches for formulation F2 with F5, F6, F8, and F10 through F12. There were a couple of formulations that had colors that matched the BY2 standard, which was F1 and F7. For F13, we didn't see a match for any of the colors. And for F14 to F24, which were the lower concentration monoclonal antibody formulations, we saw match predominantly with the color standard BY4. And we can see that for F14, F18, and F21, F22, and F24, we had very good matches with the color standard BY4. And we see that for F16, the color is somewhere between BY4 and BY3. And for F23, the color is somewhere between BY5 to BY7. So for turbidity measurement, these are the results that we obtained. So you can see that for the 210 milligram per mil solutions, that the turbidity that we measured was somewhere between 70 NTUs and approximately 170 NTUs, and that was for formulations F1 through F12. And you can see for the lower concentration formulations that the turbidity of the sample ran between 20 NTUs and approximately 300 NTUs. And you can see that the one that really stands out quite dramatically is the formulation F13. So the results and conclusion from the accelerated stability study were this. For the high concentration formulations, which were F1 through F12, those formulations were unstable and definitely showed signs of aggregation. In fact, if you remember what I mentioned when we pulled those out of the incubator at the 45 degree C 90-day time point, that those formulations had a gel-like consistency. And so also the viscosity of those formulations really increased dramatically over the course of the study, again, clearly pointing to the fact that those formulations were aggregating and that they're unstable. And so I would label those formulations as being a bad formulation, right? One that we wouldn't want to take forward. For the lower concentration formulations, the 50 milligram per mil, formulations F13, F17, F20, F22, F23, and F24 also were unstable, and those also showed signs of aggregation. And so the way that we were able to assess that was that those formulations either showed signs of particle formation, physical particle formation, or had high turbidity. And then there are formulations in the green, which were F14, F15, F16, F18 through and F21 and F19, and those clearly looked very, very good. They didn't show any signs of aggregation. The viscosities were within range, and so these formulations I labeled green because these look like they were the good formulations and that they were stable under the conditions of the study. So the summary from the accelerated stress study is that for each time point, we were able to measure the viscosity and pH of the samples using the CM Protégé bioformulation assessment system. We were able to measure viscosity, pH, color, turbidity, and particles for the very last time point, data point for the accelerated stress stability using the CM Protégé bioformulation assessment system. And the analysis of those 24 formulations took less than 1 day to do. So using the CM Protégé bioformulation system, you can analyze a very large amount of samples and get quite a bit of information about your samples in less than a day. The CM Protégé bioformulation assessment system was capable of detecting the viscosity difference that resulted in the differences from the differences in the formulation. What we showed was that the viscosity of the 50 milligram per mil formulations didn't really change during the stability study. But the viscosity of the 210 milligram formulations, you can see increased dramatically over the time, at all temperatures, and especially for the formulations that were incubated at 40 degrees C. The CM Protégé bioformulation system also detected and measured the visible particles in the low protein concentrations. And visible particles were detected post-incubation. I have here 4, but it's really 6 of the formulations. And the number of particles that were detected ranged between 1 and 20. And in conclusion, the study identified 6 formulations, F14, F15, 16, F19, F21, that were stable under the conditions that were chosen or that were tested. So in summary, the CM Protégé bioformulation system is an automated system to measure viscosity, visible particle, turbidity, color, and pH of protein vaccine formulations. And by having those analytics, it's useful to use the system to identify the optimal formulations via screening, so you can really accelerate your project turnaround times. You can analyze formulations very quickly from accelerated stability studies, which is one of the examples I tried to show. And the system integrates 5 manual assays to really streamline your workflow. And so the system has a low volume, high throughput viscosity measurement, which supports formulation screening and accelerated stability studies. It does a non-destructive analysis of particles, color, and turbidity for protein and vaccine formulations. Because we're using imaging and software analysis, it gives you an objective and consistent measurement of visible particles and color. And again, because we're using digital imaging, all images are stored for post-run reprocessing, for archiving and review. And because we're using a solid-state pH probe, we see that the pH measurements from the system are much more robust and reliable than what you would get from a glass microelectrode. And the CM Protégé bioformulation system really completely automates the measurement of viscosity, turbidity, color, visible particle measurement, and pH. And that allows you to do extended unintended operation to accelerate development, reduce your costs, and explore a broader formulation space. And more importantly, because we're freeing up scientists from doing those assays manually, we're really freeing them up so they can, from the routine laboratory work, they can actually do more productive things with their time.
Great. Thanks, Dave. That was a really informative webinar presentation, so thank you very much. So now we're going to have a short, about 10 minutes Q&A session. We've had lots of questions submitted, and so we will try to get through as many as they can, and we'll throw them at Dave and see what he can do for us. Rest assured that if you don't get your question answered now, we will follow up and answer your questions individually via email. I'd also like to say that at the conclusion of the webinar, we will keep the console open for an additional 5 minutes to give you a chance to submit any additional questions that you'd like to see answered. Okay, so first question. Dave, could you tell us a little bit more about how the particle size is measured?
So with the Freeslate visual inspection system, we actually don't measure the particle size with the technique that we use. We're just really measuring whether particles exist or not exist. And it's really useful mainly from a screening standpoint when you're trying to quickly look at your formulation to assess whether visible particles formed or not.
Great, thank you. What's the minimum sample volume needed to measure viscosity?
So the minimum sample volume that is required to measure viscosity is 100 microliters. And since we require such a small amount of sample, you really don't have to waste a lot of sample doing like you would do with other viscosity measurement systems.
Great. And what's the principle that the turbidity measurement's working on? So
the turbidity system, the way that it works is that we're really measuring well, so let me back up here. So since we're illuminating the vial from behind and we're measuring the amount of light that's being transmitted through the sample, what we're measuring is really the difference in the difference that the solution makes between measuring a blank vial and the vial that we're actually measuring, the sample vial. And so we're measuring the difference in luminescence. And so it's a little bit different than the standard method that you would use to measure turbidity, but we find that it's very effective.
Great. Could you please explain the principle for the viscosity measurement to us?
Yeah, so the way the viscometer works is that we have a stainless steel capillary tube. And so we take the solution that's in the PDT pipette, or the positive displacement pipette, and we inject it into the capillary. And at the base of the capillary, or I'm sorry, at the top of the capillary is a pressure sensor. And so what we're doing is we're measuring the pressure difference of the solution as it's flowing into the capillary. And so what I didn't mention in the presentation was prior to doing the analysis of samples, you would measure some viscosity standards to calibrate the capillary viscometer. And then based on that calibration, you then can quantitate the viscosity of your sample that you're measuring.
Great. And so following on from that, what's the maximum solution viscosity that can be used for particle counting?
Oh, that's a good question. I have to say that I have to check back because I don't recall what that is off the top of my head.
Oh, okay. So
I take that back. I should know that because I mentioned a couple times in the webinar. It's 100 CP is the maximum viscosity that we can measure with the viscometer.
Okay, great. And can you tell us the particle size limit for the analysis?
So when we were benchmarking the particle detection on the visual inspection station, we could measure particles that were really quite small, down to about 35 microns, or actually smaller than that. We detected some 1 micron particles. But in terms of reliable particle detection, what we'd set as the cutoff was any particles that are above 80 microns. So the system is really designed to do visible particle detection and not subvisible particle detection.
Thank you. Can you explain a little bit why color is an important assay?
So, color is an important assay because, when first of all, if the color is cloudy, not cloudy, but if it's really, really yellow or really, really brown, talking to customers, my understanding is that the pharmaceutical companies don't like the solutions to be that color. In addition, the way that we were doing the color analysis for the accelerated stability study was showing that the formulation started out as being clear, and as they went through the accelerated stability study, their color changed. And so that's an indication that there's some kind of change going on in that biologic, so that you need to understand what those changes are. So it's an indicator of a change happening in the biologic. Great.
Can you determine the viscosity of a slurry?
As I would think that if the slurry had a viscosity higher than 100 centipoise, that it would be beyond the range of the viscometer. But if the slurry was within the range of 100 centipoise, we could measure it.
Great. So I think we've got time for two more. When you're processing images for particle detection, why does your software subtract the consecutive image pairs?
Yeah, so the reason why we subtract the consecutive image pair is that we eliminate any kind of artifacts or any kind of defects that are present in the glass. So what happens is that, let's say you had a spot on a fingerprint, let's say, on the vial. And so if we just use the image from the vial, we would probably actually count that fingerprint as a particle or particles. And so by taking the difference image, we actually subtract out the fingerprint, so it isn't in the final difference image that we use to do the blob analysis. And so we eliminate those artifacts that are in the glass that could be miscounted as particles.
Great. Okay, so this is the last question I think we have time for today. And I really didn't write this one, so I'm going to read it because it's great. Does this system integrate with the CM Protégé PharmD system from Freeslate?
So the physical integration, it does not. But you can integrate it through, I would say, very similar to a virtual integration, where you could use the LEA software as the means where it's kind of pulling the data together so it can view it in a consolidated way.
Great. Well, Dave, thank you very much for your time and for answering those questions. Before we go, I would just like to say that we store all of our previously recorded webinars on our website at www.freeslate.com. Just select Technical Resources from the upper navigation menu, select Learning Center from the drop-down menu, and you should be able to find a list of previously recorded webinars there. So we hope you enjoyed this. We hope that you found it informative. And remember that we will be answering your unanswered questions via email. So thank you very much, and have a great day, everyone.